All right, hello everybody. Today we're going to be talking about biotechnologies, and we're just going to touch on a few of them. There are so many amazing things going on in biotechnology. I know lots of you are interested in these as careers, and we don't get to touch on some of the cool ones. We're not going to talk about cloning. We're not going to talk about stem cell research. Even just analyzing the DNA and DNA analysis, we'll talk about what that is at the very end. Even that we barely touch on. But we're going to do some cool things today. So I think you'll be super interested. I will mention there's a little trigger warning down at the bottom that says we'll look at DNA fingerprints from several rape cases. That's going to be in class. We won't be doing that during this presentation. So the trigger warning is removed for this presentation. All right, let's go ahead and get started, see what we can find out about biotechnologies. All right, first of all, we need to talk about restriction enzymes. The discovery of restriction enzymes, which are actually found in bacteria, and there are hundreds of them, was like universe changing for the scientists, the genetic scientists. Restriction enzymes, also called endonucleases, their real job in nature is to cut DNA during editing processes so that correct DNA can be placed in if there's a wrong nucleotide pair or something like that. So they cut DNA, but they identify very specific nucleotide sequences, and they can either cut the DNA straight across the two sides. So remember, you've got two strands on your DNA. The restriction enzyme can cut straight through them and create what's called a blunt cut. Or what scientists love is they can also produce something called a sticky end. And I want you to really take a look at this diagram because this comes up repeatedly with biotechnologies and it's amazing. So some of these endonucleases, when they cut, instead of cutting straight across the DNA, they cut in a staggered manner. So take a look at the diagram, the very first one where there's an arrow pointing down and then there's an arrow pointing up over here. It cuts between the G and the A on the top and the G and the A on the bottom. And then when it pulls it apart, notice that the 5' ends of the top strand and the 5' end of the bottom strand have what scientists creatively called sticky ends. And those are sections, tiny sections of single-stranded DNA. And the reason that they are important is that they're super attracted to anybody else that is single-stranded DNA that could bond with them, so make those hydrogen bonds. And so that's going to allow scientists to do all kinds of amazing things, but in particular, it's going to allow scientists to add genes of interest. And so when we talk about a gene of interest, we're talking about a gene, let's say a scientist is doing cancer research and they think they have a gene that is leading to cancer. They can cut that gene with the same restriction enzyme, and then they can cut a bacteria's plasmid with the same restriction enzyme and look at it in the diagram in gray. They're able to insert that gene of interest, maybe the cancer gene, into this bacteria's green DNA. And they stick together because of these sticky ends, because the letters, the nucleotides are attracted to each other. The opposite nucleotides are attracted to each other. And then they use a little bit of ligase and it seals it all up. And it allows us to take DNA from one source and put it in another source. Now, if that's not a little bit scary, it is, but there's super great things that can happen from it also. Okay, so restriction enzymes allow us to do DNA fingerprinting, and that's the next thing that we're going to be talking about. Each person's DNA is slightly different in terms of their patterns. So the A's, T's, C's, and G's are just a little bit different. So the enzymes are going to cut the DNA in different locations, and that's going to produce different banding patterns. We're going to see that in just a second. We can also use these restriction enzymes for the genetic engineering experiments that I just talked about. Okay, moving on. Let's talk about PCR. This is also an incredible invention. PCR uses a machine. So this is called polymerase chain reaction. And I feel like the name is very, it doesn't really explain very well what it is, but if you think about it, DNA polymerase is what copies the DNA, and it's a chain reaction. So we're just getting DNA copied and copied and copied and copied, and that's the idea here. So it's used to make bajillions of copies of DNA. So I want you to take a look at this dried drop of blood found at a crime scene. There's some red blood cells and there's some white blood cells. Red blood cells don't even have DNA. Their nucleus leaves, it disintegrates over time. So the only cells in this tiny little drop of blood that have DNA in them are the few white blood cells. Well, that's not enough DNA for any scientist to work with. They could maybe scrape it up and maybe put it in a test tube, and then what do you do? So PCR is able to take that tiny amount of DNA and copy it and copy it and copy it and copy it until you have billions of copies. And now the scientist or the forensic scientist, they have a test tube of that DNA, and now they can do some DNA fingerprinting or whatever other things they need to do. So the other thing, what we just talked about, let's say you're a cancer researcher and you've managed to isolate this one gene and you can't see it. Remember, we can't visualize any of this. All they have is this one gene and it's at the molecular level. How can they get lots of copies of it so that they have a test tube so that they can actually suck it up with a micropipette and do something with it? And that's polymerase chain reaction. We can copy it over and over and over and over again, and then we have many copies of it. All right, so how does PCR work? It's pretty ingenious. They use a machine, so it's not happening, like out in the room or something like that. So there's a machine that does this. The first thing it does is it heats up the DNA and it denatures the DNA. Those hydrogen bonds are very weak, and when there's so much energy because of the heat, they pull apart. And so you've got two separate strands. So take a look at the diagram. You've got a target sequence. They denature it, so the target sequence is now two separate strands. They're single-stranded, but they're super attracted to each other. So if we cool it down, they're just going to come right back together again. So this next section, before they cool it down, they add some primers that snap on and they prevent the two from being able to come together. So notice the primers in the diagram where it says annealing. So those primers get in the way, and the primers prevent the two strands from coming back together, and they provide a starting point for the next step. The next step is called extension, and that's at sort of a mid-level temperature. And that's where they add a very I want to say sensitive, that's the opposite. Heat tolerant, there we go. A very heat tolerant DNA polymerase. And it's called Taq polymerase. It comes from a bacteria that is a thermoacidophile. It lives in super crazy hot environments. And so it can handle higher temperatures. So then this Taq polymerase attaches the primers and replicates the DNA sequence. So look at step number 3. It just copied. So now we started with one copy. We had one copy. We separated them. We put primers on. Now we copy both of them. So we have two double-stranded sections of DNA. And now we repeat over and over and over again. So now we add heat, they separate, we add primers, we copy, and now we have 4 copies. We add heat, they separate, we add primers, we add the Taq polymerase, and they copy again. And so we're able to make billions of copies. I'm going to show you a quick video of this. It's about 1.5 minutes long, as long as they don't show us a commercial.
Polymerase chain reaction, or PCR, uses repeated cycles of heating and cooling to make many copies of a specific region of DNA. First, the temperature is raised to near boiling, causing the double-stranded DNA to separate or denature into single strands. When the temperature is decreased, short DNA sequences known as primers bind or anneal to complementary matches on the target DNA sequence. The primers bracket the target sequence to be copied. At a slightly higher temperature, the enzyme Taq polymerase, shown here in blue, binds to the primed sequences and adds nucleotides to extend the second strand. This completes the first cycle.
They heated. They're going to add primers.
Cycles, the process of denaturing, annealing, and extending are repeated to make additional DNA copies.
Heating, add the primers. There we go.
After 3 cycles, the target sequence defined by the primers begins to accumulate.
That's the gene of interest. We're getting more and more of it.
After 30 cycles, as many as 1 billion copies of the target sequence are produced from a single starting molecule.
Awesome. Okay, I don't even know how to pause that. Hopefully it won't start talking. I'll make it go away. All right, back to where we were. How do I make it present again? I don't know what I'm doing, everybody. Try to make it quick. Okay. And zip through, we just went through the different things. Okay, now let's talk DNA fingerprinting. So it has nothing to do with our fingerprints. The reason they call it DNA fingerprinting, my picture is in the way. That's an example of a DNA fingerprint. Let's see if I can point to it up there. That's an example of a DNA fingerprint. It produces a series of bands on a gel. And we call it fingerprinting for two reasons. Number one, when you have a fingerprint, it's a series of lines, and scientists are looking at those lines. And you've got lines here in this DNA fingerprint. But it's also called fingerprinting because if done correctly, each person should produce their own DNA fingerprint, so their own series of lines in the DNA fingerprint, not unlike having very unique fingerprints each person has. So it's got some similarities, but it has nothing to do with our actual fingerprints. All right, so this is how DNA fingerprinting is done. A DNA sample is cut with restriction enzymes, and the more cuts that are done, the less likely two people will produce the same pattern. So if we're trying to compare two human, there's different reasons to do gel electrophoresis. But if you're trying to compare, say, a crime scene DNA with some suspect DNA, and you want to make sure that you don't accidentally convict the wrong suspect, you want to add several restriction enzymes. With each restriction enzyme that's added, the odds that two people will produce the same DNA pattern becomes less and less. I'll make up some numbers. So with one restriction enzyme, maybe the odds are 1 in 10,000, which is really quite amazing when you think about it. But then if you add the second enzyme, maybe now it's 1 in a million. And then if you add a third enzyme, maybe it's 1 in 50 million. I made those numbers up, but that gives you the idea. The more restriction enzymes they use, the more chopped up the DNA is, the more unique patterns those enzymes identify in somebody's DNA, and the less and less likely they are to produce the same pattern. So the more cuts, the less likely two people will produce the same pattern. The cut DNA is then, we use a micropipette, is added to the wells in an agarose gel. It's got the consistency sort of like Jell-O, a little bit thicker than Jell-O. The gel is added to an electrophoresis machine, and you see that there in the diagram, and electricity is added. Now, importantly, the DNA fragments are negatively charged. So remember that like charges repel and opposite charges attract. So if if the DNA is negatively charged and we put it on the negative end of the electric current, they're gonna push away from the negative end of the electric current, and they're going to be attracted toward the positive end of the current. And so that means they're going to start migrating across the gel, and they're going to start moving toward the positive end. Smaller fragments experience less resistance as they're traveling across the gel, so they move faster, and big, cumbersome, large fragments of DNA are going to go slower and not travel as far. So after about 20 minutes, we turn off the machine. The short bands have traveled the furthest, so they're closest to the positive end, and the large fragments haven't traveled very far, and so they're closest to the negative end. Now, can you see when you look at the bands that they're big or small? Not at all. We're just looking at how far they traveled. That's what our eyeballs are identifying. So here's just a pretend one that I made up. So in the example to the left, we have two suspects, and there's DNA found at the crime scene. That DNA might be hair, it might be, say, sperm from a rape test, it could be skin, it could be blood, any number of different DNA sources. So the DNA, there's hardly any. They copy it with PCR, so now they have a test tube full. They add some restriction enzymes to cut the DNA, and hopefully they add several restriction enzymes. In this overly simplistic example, the restriction enzymes cut whenever there's a G followed by an A. So, a real human being would have 3 billion letters. I have about 50 letters here. So I'm just cutting every time there's a G followed by an A. So I'm going to let you see that. So every time, look at the left now. Every time, so there's Mr. Green is one suspect and Mr. Red is the other suspect. I don't know, I just made that up. So notice that every time there's a G and an A, the restriction enzyme is cutting the DNA. So notice that the first fragment, the two suspects are exactly the same. They have a G and an A in exactly the same place. The next cut, they're exactly the same. They both have a 5 base pair fragment, and they have a 9 base pair fragment. But then the third cut, notice where there's a number 7 on Mr. Green, he has a G and an A. But Mr. Red, he has a T instead of a G there. That's part of what makes them non-identical. They're slightly different people. So in this case, the fragment didn't, the restriction enzyme didn't cut there. So this fingerprint is going to have a really large fragment that has 15 base pairs. Large in my analogy, it's not really large. And the other one's going to have a 7 and an 8 band. So now we take a look. It says the cut DNA is then added to the gel with the micropipettes. Electricity is added, and the negatively charged DNA fragments migrate toward the positive end. So notice the negative end down here and the positive end here. After 20 minutes, the machine is shut off and the fingerprints are compared. So they moved across after 20 minutes. This is the crime scene DNA. This is the pattern from the crime scene. And then this is the pattern from suspect number 1 and the pattern from suspect number 2. Now I have numbers over here that are showing the size of the fragments. What they usually do, they don't usually have numbers. They have something that's called a ladder. I'm forgetting, there's another word for it. But they have sample DNA that they know how many base pairs it has, and it makes a ladder. And then we can compare the suspect's DNA to this ladder to know how many base pairs are involved in any one fragment. And so in my simplistic example, here's the DNA fragment that was 5. Remember, Mr. Red had a 5. And here's the one that was a 9. Mr. Red had a 9. And then he also had a 15. And there's his band at 15. Whereas Mr. Green had a 5, a 7, an 8, and a 9. And so he produces a very different DNA fingerprint. And it's definitely a match to the crime scene. And suspect 2 is absolutely eliminated as a possible person who donated that DNA. All right, gel electrophoresis can be used for other things. It can be used, obviously, for crime scene analysis. You may have heard that it was used for COVID research and research in genetic disorders. It can be used to determine paternity or relatedness in general. So we'll take a look at this DNA fingerprint to the left. The child's bands are supposed to be a combination of the mother's and the father's. Now, paternity testing is never 100%, but just take a look at this example. So the child should have gotten half of its bands from the mom and half of its bands from the dad. And so what we want to do is, we know the mom has half of them. We know the mom, because she gave birth to this child, we know that's the mom. We're trying to figure out which male is the father. And so we're trying to see which bands. So let me just walk you through it. So here's child. Mom doesn't have this band right here that the child has. Oh, but male number 1 does. Actually, and so does male number 2. So it could be either man could be his father or her father at this point. So now let's go down. This band right here came from mom. But where did this band come from? Oh, this male has it, so that's a possibility. Let's go down further. Mom does not have this band, so it must have come from father. Oh, look at that. Father has that band. This band here must have come from mother. This band came from the dad. This band came from the mom, so forth and so on. And you can see that the child is a conglomeration of the two, so male number 1 is the correct answer. Now, there are 2 websites that have actual DNA fingerprints, and they have like little multiple choice questions, and I encourage you to try them. So go do that. All right, moving on. Steps of a genetic engineering experiment. This is mind-blowing and so important in our advances in understanding DNA. The thing that you really have to get is that all living things on this planet use the same A's, T's, C's, G's, and U's. But we all use exactly the same building blocks for our DNA, which is an argument that we're all related, that we have this common ancestor that was, the original life form on this planet. But it allows us in a great way and in a really scary way to combine DNA from different sources. So in this genetic engineering experiment, it says scientists can now remove a gene of interest from one source and add it to the DNA of another source. So we're talking about actually moving DNA from one species to another species. Now, for the most part, these experiments are taking place with bacteria. You can take a gene from any organism, a human, a goat, a jellyfish that glows, that's a huge one. And you can add that DNA to a bacteria, and the bacteria can make that one protein. Remember, a gene tells you how to make a protein, that's roughly, or an enzyme, which is a type of protein. So you can add that. It's not that you're going to turn a bacteria into a human being, because you can't add the entire human genome, but you can add a single gene, and then you can study that gene and see what that gene does. And you can also ask the bacteria, because they reproduce every 20 minutes, they clone themselves. So if you get a gene successfully added into a bacteria, and then you just grow that bacteria and it reproduces and reproduces and reproduces and reproduces, it's now going to make that protein that you inserted the gene for over and over and over and over again. And that's what we've done with things like insulin. That's what we've done with human growth hormone. We've added those genes to bacteria and just let the bacteria make it for us. So these are the steps. They're super cool and amazing. Remove the gene of interest using a restriction enzyme. Now, I make that sound really easy. It's incredibly, staggeringly difficult, because remember, the scientists, they can't see the DNA. They can't see the letters A, T, C, and G. So they have to do all this background work first to figure out what are the letters of the gene of interest. How do they even know which gene is the gene that they're interested in? So there's an enormous amount of work that goes into this. But once they have a gene that they're suspect of something, or they want to learn more about, then they find a restriction enzyme that cuts the gene on either end. So you don't want to wreck the gene. You can't cut in the middle of the gene. You got to cut on the front end and the back end of the gene. And it's even more complicated than that, but we'll go with that. Then they have a plasmid, and there's actually plasmids that you can buy at the store, so to speak, scientist stores. You can order them. They're commercially available, and they're plasmids that have different things in them, and we're going to learn about them later. But you use the same restriction enzyme to create sticky enzymes in that plasmid. So look at step number 2. They have a plasmid. The same restriction enzyme is going to cut open the plasmid, and it's going to produce the same sticky ends. So they're going to be complementary. So then in step 3, you mix the gene of interest with the plasmid, and sometimes, not always, sometimes the plasmid will just close right back up on itself. But if you have the gene of interest, occasionally that plasmid will close on the gene of interest, and now you have this recombinant DNA. So you mix the DNA sample, and you use ligase to mix them and to bind them together. And then you allow the bacteria to suck up the plasmids. So you put the plasmids into an environment where there are bacteria. And remember, bacteria can do this process called transformation. That's a technical term. That's not like a transformative experience. It's a real word, transformation, where bacteria can suck up the DNA from the environment, and then they immediately possess whatever characteristics are on that plasmid. So they suck up the recombinant plasmid, and now they start producing whatever that gene of interest tells them to produce. So it says the bacteria will now produce the protein coded for by the new DNA. All right, so examples of recombinant DNA that we have in our normal society now. Scientists are figuring out ways to add genes for pest resistance to plants, so that they're less likely, like when farmers are growing plants, they're less likely to be eaten by certain organisms that can kill the plants. Genes are added to bacteria to clean up, have been added to bacteria to clean up toxic waste spills. We've used recombinant engineering to create proteins that dissolve blood clots for people. And we've, as I mentioned earlier, we've even mass-produced human growth hormone that then we can give to people whose growth is small or growth is stunted for some reason, and help them to reach a more natural height than the stunted growth that they had. All right, and then the very last thing we're going to talk about, I'm not going to give a whole lot of detail about DNA sequencing. It's fascinating, super, super cool. It's the process of determining the sequence of nucleotides, the A's, T's, C's, and G's in a piece of DNA. The Human Genome Project was the first big effort at doing an entire organism. So that lasted from October of 1990 until April of 2003. They thought it was going to take decades, and they did it extremely quickly because The technology kept advancing while they were working on the project, and so they made it through the entire 3 billion sets of A's, T's, C's, and G's in humans. So their goal was determining the base pairs that make up human DNA, and then of identifying, mapping, and sequencing all of the genes of the human genome. We are still looking at what they discovered and the genes. We're still using all that information all over the world. So it says here, this is a quote. Today, with the right equipment and materials, sequencing a short piece of DNA is relatively straightforward. Sequencing an entire genome, which is all of an organism's DNA, remains a really complex task. It requires breaking the DNA of the genome into many smaller pieces, sequencing the pieces, and assembling the sequences into a single long consensus, a line that overlaps. That's all I can say. However, thanks to new methods that have been developed over the past 2 decades, genome sequencing is now much faster and less expensive than it was during the Human Genome Project. We're making constant steps forward. So I'm just going to show you this really quickly. You don't need to know anything about it, but I just want you to be aware of it. This is called the Sanger method. The Sanger method is how they are identifying, and it has to do with this pretty colorful picture there also, how they're identifying A's, T's, C's, and G's. They use lasers, and the lasers identify whether something is a G, T, C, or A. You don't need to know really anything about it, but just be aware that DNA sequencing is something that we're working on and getting a lot more information about our DNA that way. All right, ladies and gentlemen, that is it for biotechnology. Let me know if you have any questions.